Welcome to the Biochemistry Academy, where we present information in the simplest way for anyone to understand. Today we'll talk about the structure of protein. Remember, we say proteins are the polymers of L-amino acid, and protein plays so much role in our body, like making up channels for certain molecule to pass, or they can act as enzyme to speed up our chemical processes in the body, and many more. Now, the human body is always making protein through a process called protein synthesis. Proteins do fold to carry out certain function. Now, to understand this, we are going to talk about the different levels of protein structure. Proteins have a primary, secondary, tertiary, and a quaternary structure. Now, the first level is the primary structure of protein. The primary structure of protein is a linear sequence of amino acids that make up a protein. Remember, I said that amino acids are the monomers, or they are the building blocks of protein, and they are held together by a peptide bond. Now, the linear sequence is either written in a 3-letter or a 1-letter form. An example looks like this. Now, the primary structure of protein is always written from the N-terminal of the first amino acid to the C-terminal of the last amino acid. The information of the primary structure of protein can be obtained by sequencing the gene that encodes the protein. In the secondary structure of protein is where folding begins to start. Now, the sequence of amino acid mentioned in the primary structure can fold in different ways. The most common ways are the α-helix and the beta-plated sheet. Note that the type of folding depends on the amino acid arrangement. And if you remember, we said that these amino acids are held together by a peptide bond. Now, the peptide bond has a carbon that is attached to oxygen and the nitrogen that is attached to hydrogen. Now, both the oxygen and the hydrogen have the ability to form a hydrogen bond, which then results in the folding of peptide chains, which can either be the α-helix or the beta-plated sheet. Now, the α-helix is the most common secondary structure of protein. In this structure, the protein attains a spiral structure such that the side chain of the amino acid remains outward. The α-helix is stabilized by this hydrogen bond. It is formed between the hydrogen of the nitrogen and the oxygen that is attached to the carbon. Now, individual hydrogen bonds are weak, but collectively they are strong enough to stabilize the spiral structure. She notes that all the peptide bonds except the first and the last participate in hydrogen bonding. The beta-plated sheet, which is the secondary structure of protein. Now, in the α-helix, the hydrogen bonding was between the same peptide chain, while in the beta-plated sheet, the hydrogen bonding is formed between two different polypeptide chains or the same polypeptide chain. The beta-plated sheets can either be arranged in a parallel or an antiparallel fashion. Next is the tertiary structure of protein, where more folding occurs in a 3-dimensional shape of a functional protein, and this is due to the R groups, which are called the side chains. Remember, in our last video we said that the outgroups found in amino acid can vary among different amino acid, meaning that the outgroup can define the amino acid and can make amino acid behave a certain way. For example, some outgroup can be hydrophilic, which means that they like water, or they can be hydrophobic, which means they don't like water. Now, when folding is going on, the amino acid with the hydrophilic group hang on the outside, while the hydrophobic out group hang in the inside of the protein. Now, the 3D shape is also due to other interaction besides the hydrophobic and hydrophilic interaction. Now, this other interaction includes van der Waals interaction, or ionic bonds, or disulfide bonds, or also hydrogen bonds, which are all involved in the outgroup, which then cause the folding of the tertiary structure of proteins. Next is the quaternary structure of protein. Now, the tertiary structure of protein that was formed can further interact with other tertiary structure of protein to either form dimers, trimers, tetramers, and so on. Now, the structure which is then formed is called quaternary structure. Now, most of the protein in our cells have the quaternary structure. If this video have helped you, please don't forget to like, share, and subscribe to the Biochemistry Academy for more content like this. Arigato.